Thank you all for standing by and welcome to our webinar entitled Microplastics Removal During Drinking Water Treatment. This webinar series called Freshwater Science will highlight Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus from human health and fish farming to harmful algal blooms and human decision-making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. John Lenhart from The Ohio State University. Dr. Lenhart is a professor of environmental engineering and associate chair in the Department of Civil, Environmental, and Geodetic Engineering. He is also the co-director of the Ohio Water Resources Center. He received his PhD in environmental science and in engineering from the Colorado School of Mines. He followed that up with a National Research Council postdoc fellowship at the U.S. Geological Survey and a second postdoc position at Yale. He joined the faculty of Ohio State University in 2003, and his research interests are in environmental aquatic chemistry. He has authored or co-authored more than 100 publications and is the recipient of a National Science Foundation Career Award. We're really delighted to have Dr. Lenhart here today to discuss his research. But before we do that, just a few things about the webinar itself. All participants will be in a listen-only mode. Afterwards, we'll conduct a question and answer session. If you have any questions during this time, please feel free to pull up that chat feature and ask away, and we will get to as many questions as possible. As a reminder, this webinar is being auto-captioned and is being recorded for posting on our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It will help us continue to give you great webinars. So without any further delay, I'd like to introduce Dr. John Lenhart presenting microplastic removal during drinking water treatment. Dr. Lenhart.
Thank you very much, Jill. I'm going to try to share here real quick.
All right.
All right. Hopefully that works for everybody. So I'd like to start out by acknowledging the student who's actually done most of the work on this project, Megan Jamison. She's a PhD student at Ohio State in the Environmental Science Graduate Program. I'd like to acknowledge the support of Sea Grant for providing funding for this. And as I'm going through this presentation, one thing I want everybody to keep in mind that this is still research in progress. We've only been doing this for a little over a year now. So much of what we're reporting is still being kind of finalized. This is not a final story. Just keep that in mind. All right, let's start with a little bit of background. We think about microplastics. First off, we should think about the generation of plastic waste as a whole, or just the use of plastics. And this diagram comes from a compendium from the United Nations that they put out, I think about on an every year basis, that focuses on different environmental concerns. And one of those, as of late, has been microplastics, originally from the perspective of marine systems, but over the last 10 years or so, more interest has been put towards freshwater, surface water systems. And so, as can be seen in this diagram, initial commercial availability of plastics was around 1950. And since that point in time, the amount of plastics that have been generated has increased. And in fact, if you were to plot this data, you would find that it's still following an exponential increase as of now. To the right of this white line, these are all projected numbers. This is what would be anticipated to occur if the current generation proceeds. One thing I find interesting is this other portion of the diagram that shows how the plastics have ended up over this period of time. So over the last 67 years, roughly 9,200 million tons of plastics have been generated. Of those, about 2,900 million tons are still being used. Around 1 million tons have been incinerated. It's kind of shaded out here. About 700 million tons have been recycled, and quite a bit, more than half, has been discarded in some way. So some of that's going to a landfill, and some of that is being discarded into the environment. A couple other points to make is the fact that roughly half of the plastics that we've used have been generated over the last 20 years, and half of the plastics that we're using are only used once. So microplastics is a plastics management related issue. So if we look at kind of defining the different categories of plastics, there's a lot of terms that go around, and they all have a bearing on the size of the plastic particle itself. So we can start with something that's a megaplastic, which is big, a dog-sized piece of plastic, macro, meso, micro, nano. So microplastics are smaller or roughly the same size as a dust mite. And then a nano, we're talking very small, bacteria or virus size. So overall, kind of the micro to nano is the area of interest and of interest from my perspective and a public health interest. So depending upon where you look at for the definition, the size discrimination for microplastics is 5 millimeters or less. This diagram shows 1 millimeter, so it's 1 or 5 and smaller. Different categories of microplastics. So microplastics can be coming from primary sources or secondary source. So the primary sources are shown on the top here. And so these are small sized plastics that are specifically prepared at that small size and used commercially at that size. So in things like medical products, abrasives, personal care products. Secondary microplastics are those that are typically generated from the breakdown of larger size plastics. And so they would come from a variety of different sources and again, are just breakdown products. In terms of surface water environments, the primary sources are coming from runoff, surface runoff, as well as from effluent of wastewater treatment plants. A fair amount of microplastics are removed in wastewater treatment, but not all of them. So common microplastics are shown here in this table. I'm not going to go through this exhaustively, but we have high density, low density polyethylene, polypropylene, polystyrene, common plastic types. You can see here what they're used for. So polyethylene, shopping bags, plastic bottles, garbage bags, composite lumber, polypropylene, shopping bags, camping gear, so forth and so on. Of interest is the density of these materials. So some plastics have a lower density than water and some have a higher density than water. So some of the plastics will float and some will not. And that's important for understanding where the plastics are going to go. From our perspective, it's important to understand this because we need to try to extract these from the samples that we're analyzing. In terms of some other categories that are used, you can look at plastics that are fragments. So this image shows a fragment of a plastic particle. There are fibers, and so this image shows a fiber. And then beads. So beads would often be particles that were actually generated at that size. So that would be a primary microplastic particle. It's been in the media quite a bit, so run through some of the recent headlines. And so this is coming from the BBC. Microplastics are everywhere. Is it possible to reduce our exposure? Tap water. So this is what we're of interest, and this comes from Forbes. So there's investment, economic interest in this. A report came out, or actually not a report, an article was released about a week and a half ago looking at nanoplastics in bottled water. So that got a lot of media attention. Of particular concern to me is the evidence of microplastics in beer. As a frequent consumer of beer, this is very concerning. I can't imagine what we're going to do if we find plastics in beer. But that being said, public health. So why do we care as environmental engineers, environmental scientists, public health folks, is the potential for public health concern. So microplastics have both primary and secondary toxic effects potential. Again, this is all preliminary evidence that's out there right now. There is no definitive proof that these things are causing any sort of health effects that I'm aware of. But again, this is not my area of expertise. But upon exposure to microplastics, what can occur is if they are ingested, they can be transported into the circulatory system. The very small plastics can get absorbed into the body and transported through the body. And so they could actually end up going in other organs, possibly leading to liver toxicity. They have been shown to pass into through the berry into the placental fluid, so they could get passed on to the fetus. And recently, it's been shown that they can actually penetrate the blood-brain barrier, so it's potential that they could get accumulated within the brain and have potential effects there. Other issues are associated with the fact that the plastics themselves during their production may have other constituents associated with them. So additives such as plasticizers or flame retardants or dyes, those could get leached out and they may have potential health effects. Plastics are also good at accumulating contaminants in the environment. So various organic contaminants such as PFAS or pesticides could be accumulated by the microplastics and could potentially be released if they were ingested. They can also take up heavy metals as well. In terms of the ability to do this, it increases as the plastics get smaller. So nanoplastics have a higher surface area, so their ability to accumulate contaminants would be expected to be greater. Our particular research is focused on looking at removal in drinking water treatment, specifically conventional drinking water treatment. So conventional drinking water treatment would be what most major utilities are utilizing. And it's comprised of a series of processes illustrated here. So they would use a coagulation process where they add a chemical to cause particles to stick together. They use a flocculation process that gently mixes that solution to get those particles to aggregate and form bigger particles. And then those particles settle out, and then they go through filtration, and that's a final polishing step to try to remove whatever residual particles might be remaining. And then disinfection, and it is passed on to the consumer. In terms of what is known, so there have been some studies looking at drinking water and microplastics in those. Not a lot. What is fairly well understood is that the microplastics greater than 10 microns are going to be removed pretty well. Less is known about microplastics less than 10 microns. And what has been published is mostly associated with those that are greater than 2.5 microns. Almost no existing results are looking at particles less than 1 micron. There are several studies that have looked at bench scale approaches looking at particle removal, almost exclusively using model plastics, which are quite a bit different than environmental plastics. And for our particular interest, the idea that configuration of treatment, how that impacts things, that's not well understood. So that gets us to our objectives. So again, we're looking at specifically trying to understand removal during conventional drinking water treatment. We're interested in trying to understand how the configuration of the treatment systems are influencing removal. And we are particularly focused at the smaller size plastics because those are the ones that are less well understood and also exhibit the greatest potential to have public health effects. So the reason why we're thinking about looking at different configurations, so in coagulation, the chemical that's used could be different. That might have some sort of an impact. During flocculation, the design for flocculation could be different. That might have an impact. Sedimentation, there are different ways to get particles to settle out. That might have an impact. And then in filtration, different media is used. The depth of the media used, it might be different, and that might have an effect as well. And then also disinfection. So disinfection might have an effect because different disinfectants might break up bigger particles to form smaller particles to a greater extent. Okay, just real quick. So we have sampled from 4 different sites. These are associated with water treatment plants that use Lake Erie. Each of these uses a different chemical coagulant, but they're all using an aluminum-based chemical coagulant. So we have not worked with any plants that are using an iron-based coagulant. They use different configurations of flocculation and sedimentation. They're all using rapid granular media filtration, but different media, different depths of media, and disinfection processes are different. So we're trying to get a spectrum of different processes. They're all different sized plants as well. We have collected raw and treated water. We've also collected water in between unit processes, but we haven't analyzed all that. So I'll just be looking at raw and treated water today. We try our best to avoid contaminating our samples with ambient and ever-present plastics. So we use clean methods as much as possible. We avoid using plastic or any other synthetics in the laboratory spaces, use glass and metal for any sort of material handling. Just real quick in terms of how we isolate the plastic. So we collect the samples in, again, a glass jar. We do an oxidation step if the sample is coming from the lake because we need to oxidize the organic matter associated with that to remove it because that interferes with subsequent processing. We do density separation to get the low density plastics. That allows us, so they float or be accumulated at the top of the water. So we can do a density separation using a two-chamber separator. We then change the density of the water to then sort of isolate the more dense microplastics, so 1.3, 1.2, 1.4 kilograms per liter. Those we collect and add those to the system with our low dense plastics. We go through and filter this and collect it on an aluminum oxide filter. Methods that we're using, so we're using scanning electron microscopy. This gets us the size, number, and shape of the microplastics. And then to do specific enumeration, we're trying to develop an image analysis approach using a software called MyPAR. That's this image here on the lower right. So we're training the model so that it can actually automatically identify the plastics and then do the counting itself, because visually doing it's very laborious. So image on the right is the model's perspective of the microplastics that are in image on the top. It does a pretty good job. Avoids identifying a feature like this, which is actually a defect in the membrane. It's not a plastic, so that does not show up down there. So that makes us pretty happy. The other technique that we're using allows us to identify the actual polymer type, and this is called optical photothermal infrared spectroscopy. The advantage of this is that it allows us to identify smaller size plastics than other techniques. Other techniques that people have used are infrared, FTIR, Raman spectroscopy, and those are nominally good for particles greater than 10 microns or 5 microns. The technique that we're using allows us to identify plastic types for particles greater than a micron, so 500 nanometers. So we're looking at nanoparticles specifically. So for example, it's hard to see optically, but in here, there's a small particle that we've identified to be polyethylene. The other way we can use this technique is in a scanning mode. And so we can identify a particular wave number that's associated with a vibrational feature of plastics. So here we have CH2 stretching, and the image shows particles that are very strong in terms of that feature, as evidenced by the yellow or red, indicating an intense feature. So this we can use to identify the plastics, and then we can collect a spectrum on it. And we can also get sizing from this as well. All right, get some results. So again, we've collected treated and raw water from 4 different plants. So these results are summarized here. We have on our y-axis concentrations of our particles in terms of particles per liter. We have our 4 different plants on the bottom, and then our control over here. So we're looking at raw water and treated water. The blue is the portion that are nanoplastics. So these are plastics that we've identified as having a size less than 1 micron. The red is the microplastics, which for us is particles between 40 and 1 micron. We did not identify any particles greater than 40 microns in any of our samples. Take-home message from this particular set of results is that the overall raw water from Lake Erie roughly has about 100,000 particles per liter, and there's a fair amount of nanoplastics in the raw water. We see good removal in the treated water, and in the treated water, we see that the proportion of the particles that are nanoplastic increases, at least in terms of the particle concentration. One thing to note is in the treated water, in all cases, this water has had a turbidity of less than 0.1 NTU. So this is, again, treated water, and it's meeting spec. The other way you can look at the data is in terms of the mass concentration. So that's what's shown here. So here we've converted the particle concentration to a mass. And so it's important to note that those particle concentrations, 100,000 particles or whatever, that is then related to a mass concentration that's in the nanograms per liter. So we're small masses of particles, even though it seems like there's thousands of them, the actual mass is very, very small. So when we look at the mass, what we can see is that there's quite a bit of difference in the mass concentrations within these different samples. And this particular sample from the drinking water plant 3 had a significant amount of microplastics at the time when we were collecting these samples. I forgot to mention this in the prior slide, but there is a pretty good correlation between particle concentration and turbidity. There is no correlation between the mass concentration and turbidity. And again, in our treated water, we see good removal And in all cases, the actual concentration that we see in the treated water is less than 20 nanograms per liter. So still very small concentration in terms of the mass of plastics in the treated water. Look a little bit more closely comparing the concentrations in terms of mass and particle concentrations for the plants. I should know that plant 1, plant 2, and plant 4 had very similar results. So it's only showing plant 1 and plant 2. And then plant 3 had some different results. So that's why it's shown up here as well. So again, this is percent of the total plastics, representing it as particle count versus particle mass. Colors refer to different sizes. So the gray is our particles that are greater than 5.2 microns. The orange are the particles between 1 and 5 microns. And then the blue, those are our true nanoparticles. So you can see when you look at concentration based upon the particle count, the particle concentration, that it skews towards the nanoplastics. So there's a lot of very small particles in this case. And when you look at it in terms of the mass, the larger mass associated with the fewer, bigger particles then changes the distribution of the particles. So that's why the color change shifts, because the larger microplastics, their larger mass then changes the proportion of the overall plastic distribution. We look then at the treated water, and the treated water kind of shows similar trends in terms of the particle concentrations, in terms of the counts of particles being dominated by the nano and microplastics, and then a small signal associated with the presence of the larger size plastics when you look at it in terms of mass. So in most instances, there's very few larger plastics getting through treatment. And of those, they're on the order of 5, 6, 7, 8, 9 microns, very small. And we're talking just a handful of these particles that are present, but their larger masses then changes the distribution in terms of the percent of the total. So summarize all these results here. You can see for the 4 plants have depicted the total removal and then the nanoplastic removal. Top is based on the particle concentration. Bottom is based upon the particle mass. So we see removal overall anywhere from 50 or so to 90% based on particle concentration, and similar percent removals based upon particle mass. The removal based on particle concentration is maybe a little bit more consistent than it is in particle mass. We see a little bit more variation. In terms of the nanoplastics, for the most part, we see that it sort of tracks pretty well with the overall removal. So the plants are moving roughly all plastic types equally well when you're looking at particle concentration. Slight difference when you're looking at particle mass, because in basis of mass, the plants are removing the larger plastics, the mass to a greater extent than the smaller. So in some instances, we see on a mass basis that only 31% of the nanoplastics were removed. Last bit of results here. This is some of our results looking at the different plastic types that we've identified. And this is a compilation of raw and treated water. And so what we see is that the particles are dominated by 3 particle types. So we see polypropylene, that's the blue up here. We see polyethylene and derivatives of polyethylene, that's this green, 24%. One thing that we see that we're still struggling with, because it hasn't been reported very much, is a fair amount of synthetic rubbers. So we're still trying to identify if indeed that's what we're seeing, or maybe if it's something else and just the database we're using doesn't have the appropriate model compounds for us to identify. We see some polyesters and then a number of other types in smaller concentrations. Just to sort of summarize again, so for both our treated and raw water samples, we see a fair amount of nanoplastics and microplastics in those samples. So smaller sizes that we're able to see that others haven't been able to see provides evidence for those. The synthetic rubber that I just mentioned is not something that's widely reported in the literature, so this is something we need to investigate more. Overall removal varied to some extent for the different treatment configurations. We're still trying to identify why exactly that is. Consistent with the literature, we saw pretty good removal of the larger size microplastics. So those would be 5 microns or greater. The smaller ones was a little bit lower than the larger microplastics, but still comparable, which was something we weren't necessarily expecting. Things to consider. So there are some limitations again. So this is ongoing work. Our sample sizes are limited and some of our results are preliminary. We're still doing additional analyses and some of what we're reporting might change as we get more data. Certainly would be nice to get more systems with greater configuration variations. We've been trying to get funding to do this, but have not been successful as of yet. We're still analyzing samples where we've collected within facilities to try to identify specific configurations that are working better than other configuration. And in terms of the identifying of the plastics, one of the things that's important to note is that environmental plastics are not the same as model plastics. And so spectrum that's collected for a model plastic may not compare to an environmental plastic because the IR features can shift once the plastic is exposed to environmental conditions. And so that's something we're still struggling with. Finalize with some additional acknowledgements. So again, Ohio Sea Grant, National Science Foundation for their support of an MRI grant, which provided access to the OPTIR instrument, access to instrumentation at the Center for Electron Microscopy and Analysis, Center for Chemical and Biophysical Dynamics. There's been a couple of undergraduate students that have helped us out with this, Celia and Mia. And then there's Megan presenting a poster at an ACS meeting. And so she's the one who's actually been doing all the hard work. And this is her effort more so than mine. So I am now free to answer questions if there are any. I think I left some time.
All right. Well, we've gotten some really questions during your presentation, so let me ask as many as I can. And all right. So one of the questions that people had, and it was towards the end of your presentation, you talked about the synthetic rubber. And so several people were asking, would that be potentially rubber from tire erosion?
We're trying to figure that out. So we
Okay.
hadn't been looking a lot at the tire wear literature to see what that's producing. Our initial evaluation of that is it's not consistent with what people are reporting as tire wear particles. But again, when people report on tire wear particles, they're not looking at particles that are this small. And so it's not clear what might happen over time as these particles are in the environment, getting exposed to light and other sorts of processes that might change them. So we don't know.
Okay.
Trying to figure that out.
All right. Another question that we had several people asking whether or not any of the plastics added potentially was due to the treatment process itself, like if there was plastic in the filters or part of the filter process.
That's a good question. And that's actually something we're trying to tease out by identifying the polymer types in the plastics themselves. So if we can identify the distribution of the different polymers in the raw water and the treated water, and we can correlate which plastics are coming from the lake and which plastics are in the treated water, we can see if there's anything being introduced during treatment. So we don't know that as of yet.
Okay. Um, do you and I'm not sure, let me know if you would know this. Why are filtration systems aluminum oxide more often than iron?
Um, so that depends upon, I may be off base here, but if you're using an aluminum oxide, you can make a ceramic membrane out of that. And so some membranes might be made out of aluminum oxide. And for the most part, iron oxide is not a good substrate for making a membrane. So that might be one reason why they see aluminum oxide, because it can be used to make a ceramic, and ceramics are used in membrane processes. But again, I might be not really answering the question.
Okay. Another question we had is the raw water only coming from sewer system or also from stormwater systems?
So the raw water is collected straight from the lake. So that's coming from
Okay.
Lake Erie. So it has whatever's going into Lake Erie is what's coming into our raw water. So we were able to collect that from the intake at the plants that we're working with.
Okay.
So maybe I guess I did not make that point clear.
Okay. Um, Sorry, I'm like, are any specific plastics getting removed better than others due to maybe filter interactions?
That's one of our questions, and we don't have that figured out yet, but we're hoping to figure it out. And we're also trying to figure out the different density types. You know, are the more dense particles getting removed greater to a greater extent than the low dense? So those are all questions we're trying to answer ourselves.
Okay. I might have a few more of these.
Oh,
Sorry.
that's cool.
Um, do you know, because you kept saying this is prelim, so do you know where the plastic from shotgun wads fall in the plastic type spectrum?
I am not a gun person, so
Okay.
I'm sorry, I'm not going to be able to answer that because I'm not sure what kind of plastics are used there.
Okay. Um, sorry. Do you have data on the predominant types of microplastics, whether it's fiber, fragments, etc.?
Most of ours are fragments. We don't see very many
Okay.
fibers. In fact,
Okay.
we see very few fibers.
Okay.
But I don't have that quantified yet. I just know that that's what we're seeing.
Okay. Did any of the plants observed use UV for sterilization? And does UV light potentially alter the plastic types observed?
No, none of them are using UV. They're using pretty conventional disinfection, chemical disinfection. One's using ozone.
Okay. Could you talk a little bit about why it matters comparing the particle versus the mass distribution?
Um, so I mean, I think this is just a different way to look at the data. So the idea that there's, say, you know, 10,000 particles, that's maybe hard to get your mind around. Cause you can't see any of these particles in the water. They're all so small you can't see them. And somebody says, "Oh, there's 10,000 particles in this liter of water." Um, what does that mean? And so the other way to look at it is in terms of the actual mass of those particles. So if those 10,000 particles are very, very small, then their mass is correspondingly small. So that's why I wanted to report both the particle concentration and the mass concentration, because in most instances, the what we're seeing in the treated water is a concentration of less than 20 nanograms per liter. And so then the question for the public health people or other people that are looking at it in terms of concern for public health is that level of plastic. I mean, do we care, right? That's not a question I can answer, but if we know sort of what the concentration is in terms of the mass, I think that will help us out, because I think the particle concentration, it's maybe not terribly useful
Okay.
because they're different sizes, and you know, if you're trying to understand effects, I'm not sure that that's the most useful way to express the concentration.
Okay. Um, I have a couple more questions. We're past 12:30, but I wanted to get a couple more in if you are game to that.
Sure, sure.
All right. Um, do you think more frequent backwashing of initial filters might increase removal?
So again, we haven't teased out how well the different filtration systems are removing the microplastics, but and we also don't know specifically the mechanism for removal to understand if the backwashing would help or not.
Okay.
So I'm sorry, I'm not helping with a lot of these more focused questions.
Well, and I keep on giving them to you, so
Well, it's okay.
I'm
Hey, if
sorry.
people have money, we're happy to do the research. That's a deal.
Um, there you go.
Yeah.
And another question, we've gotten this several times, so I wanted to make sure to ask. Um, First, we had several people asking if how would people be able to find out what their particle concentration was, like say in a lake in New York? Do you know if there's like commercial labs or anything that would be willing to do that kind of thing? And then
No, no,
also
that's
asking if your lab would.
oh, uh, I mean, it's a lot of work to try to get the information that we have, and um, in terms of like having access to a scanning electron microscope, no commercial labs aren't going to have that. So the results we are collecting are very specialized. It's not a routine test that people would be able to do. I do know that there are efforts to try to identify methodology that's more routine, but that's only going to be applied to the larger plastic sizes. So 20 or 50 microns or larger. So those could be more routinely analyzed. But those, at least based upon what we've seen, are not getting into tap water. They're getting removed during treatment. So the approaches that are being developed are not really going to get at the very small particles. So that's going to require somebody smarter than me to figure out how to automate that.
Okay. The other question that several people asked, and this might be out of your scope here in general, but people were wondering what they could potentially use at home if you had any suggestions on for tap water.
I mean, I think the only approach that would probably be effective would be a reverse osmosis membrane or something similar in terms of its pore size, because the most household systems, like in your refrigerator or whatever, they'll be using a filtration media, not that much different. I mean, somewhat different, but it's going to be a filtration media. You need a pressure membrane approach. So a reverse osmosis would probably do it if you're concerned about these concentrations.
All right. I think that is the bulk of, there were still a few more, but I think we got the bulk of the questions. One question I did want to ask was if, in terms of where you are in your research, when you thought you would have, we had several people asking when you thought you would have your final results.
Oh, we're still hoping to get um, kind of things tied up and get some publications out this summer or early this fall. That's kind of the plan. And then move on to sort of the next phase. We have a bunch of other questions we're trying to pursue. But for this part, at least identifying, overall during treatment, what's happening and which plastic types are being removed, that's something we're trying to finalize over the next 6 months.
All right. All right. Well, I will end it with that. I want to thank again, Dr. Lenhart. Thank you again for your willingness to talk to us about your research. This was really an excellent discussion and very timely given what we've been seeing in the news. So this was great. I did want to remind everyone that the survey URL in the web for this webinar is in the chat feature. So please take a few minutes if you could to fill that out. Also, this webinar series is sponsored by Ohio Sea Grant and will continue on in February with John Witter from Ohio State talking about alternatives to removing nutrients and sediment using drainage. The registration link is in the chat feature, so feel free to register for that. Again, thank you, Dr. Lenhart. As always, it's always a great presentation, so we really appreciate you taking the time to talk with us today. Also, thank you to the participants for attending. We hope this was beneficial, and we hope that you'll join us again in an upcoming webinar. Thank you and have a great afternoon. Thanks again, Dr. Lenhart.